Hello, this is Stephen Wyka, and this is my public defense seminar for my PhD in plant pathology from the Department of Bioagricultural Sciences and Pest Management at Colorado State University. Today we'll be going over the lifestyle and evolution of Claviceps purpurea, the ergot fungus. But first, I would like to thank my committee members for making this all possible. To begin, I'd like to go over a brief history of the genus Claviceps, or ergot fungi, which are fungal plant pathogens of grasses and cereal crops. From 1350 BCE to the Middle Ages, ingestion of contaminated grain often led to symptoms of gangrene and burning feelings of the skin and blood vessels, which is more commonly known today as St. Anthony's fire. Despite its toxicity to humans, in the 1400s to 1800s, ergot was often prescribed at low doses to help accelerate childbirth or even induce abortions. In the 1600s, ergot was so prevalent on grasses that the early botanical drawings of rye often contained ergot, as it was thought to be a part of the plant. In 1900s, Albert Hofmann synthesized LSD from ergot alkaloids. These findings have hallucinogenic effects from the ingestion of ergot, as well as the symptoms of gangrenous, often led to debate on whether contaminated grain caused many of the witch trials around the world. Presently, ergot alkaloids are still studied in pharmacology for their therapeutic applications. However, ingestion of contaminated grain still poses a toxic health concern to humans and livestock. To mitigate these concerns, governments have applied strict regulations on the amount of ergot allowed in grain, where harvests that contain greater than 0.1% of ergot by net weight are often rejected for consumption. Despite its long research history, control of ergot still proves challenging. Claviceps species are strict ovarian parasites of grasses, meaning that they solely colonize the ovaries and do not grow systemically throughout the plant. The life cycle begins as an overwintering sclerotia in the soil, where upon favorable weather in the spring produces fruiting bodies that develop and produce ascospores, which are the primary infection of unpollinated florets in grasses. Claviceps then starts a secondary cycle where it produces asexual spores that are dispersed by rain or insects for continual infection throughout the summer. These infections then mature throughout the fall and produce mature sclerotia, which result in a replacement of a seed and thus lower the yield of cereal crop harvests. These sclerotia are then either picked up by harvesters or fall to the ground to start the cycle again. For control methods, currently we can try to reduce the amount of sclerotia in the soil through crop rotations, deep tillering, or field burnings. In addition, limiting overhead irrigation can help reduce the secondary spread of asexual spores. Furthermore, disease severity can be reduced through the use of resistant crop varieties or fungicide applications. However, currently there are no resistant crop varieties known for ergot, and fungicide applications have been shown to be not economically practical. Lastly, we can try to reduce the amount of primary infection by cutting wild grasses surrounding barley or cereal crop fields to help limit the amount of sclerotia produced. However, this method is often overlooked by growers. One of the more well-known species of Claviceps, Claviceps purpurea, has the ability to infect up to 400 different grass species, with no host specificity, no specific races, and no genetic resistance identified. This has led to the debate about whether Claviceps purpurea is actually more of a pathogen or a symbiont in nature, as some researchers have identified negative to positive effects of infection on host seed production. Where at higher infection rates, we generally see larger losses of host seed production. However, at lower infection rates, hosts can also be seen to have more seed, which are also heavier. However, these observations are from field surveys, and this has not been experimentally tested in the greenhouse. In addition, researchers have identified grazing avoidance of grasses infected with ergot, which indicates a conditional mutualistic relationship between the fungus and its hosts, indicating that infected grasses have higher fitness potential as they are not being grazed. These observations reflect ambiguity surrounding the epidemiology, lifestyle, evolution, and adaptability of Claviceps species. Therefore, my overall goal was to utilize a multidisciplinary approach to gain insight into the evolutionary and lifestyle history of Claviceps purpurea and the Claviceps genus. We achieved this through a field aspect to examine the role plant community compositions play in the epidemiology of ergot outbreaks. In addition, we utilized a greenhouse aspect to elucidate the effects that infections of Claviceps purpurea have on its host through greenhouse inoculations. And lastly, a genomics aspect where we sequenced and annotated 50 Claviceps genomes to characterize the genomic architecture and identify factors associated with the evolution of the genus, and to construct the Claviceps purpurea pangenome in positive selection landscape. For Objective 1, we utilized the barley growing region of the San Luis Valley, which is located in south-central Colorado. These surveys were conducted in 2017, 18, and 19 during the months of July, August, and September to assess ergot disease incidence on wild grasses along ditch banks adjacent to barley fields. We utilized 12 sites per time period, where sites were rated based on the history of disease ergot within the barley field. Our findings have indicated that sites with prior ergot history saw increased levels of ergot disease incidence along ditch banks in comparison to sites that had no ergot history within the barley field. We also saw increased levels of ergot incidence throughout the summer, which is concordant with the polycyclic life cycle of Claviceps species. Observations of plant community composition along ditch banks revealed that severe ergot history sites had ditch banks that were largely composed of brome species, whereas sites that had no ergot history had ditch banks that were largely composed of non-host species. In addition, we found no significant differences between the abundance of other grass species or wheatgrass species in ditch banks between the different site ratings. Furthermore, we found that brome grass species, or the abundance of brome, was correlated with disease incidence throughout the summer, as this was not the case for wheatgrass or other grass species. In addition, we found that brome grass species had the highest number of sclerotia per inflorescence, with some inflorescences containing upwards of 80 sclerotia. These results indicate that brome grass species represent large inoculum reservoirs along ditch banks in the San Luis Valley. We estimate that brome grass species can produce upwards of 68 sclerotia per meter squared at some of these severe ergot sites. This is even true in low rainfall years, as in 2017 and 2018, the San Luis Valley saw below average precipitation, indicating that these large inoculum reservoirs can be built and are maintained despite heavy rain. Moving on to Objective 2, we looked at the impact of Claviceps purpurea infection on host physiology. To do this, we utilized 2 host species, barley and wheat, with 3 experimental replications per host species and around 240 plants per trial. Plants were syringe inoculated with Claviceps purpurea at an infection rate of 0, 5, 15, or 25% of the primary tiller. Plants were blocked by inoculation date with 20 plants per block. Each of the 4 infection rates were randomized 5 times within each block. At host plant maturity, plants were harvested and measurements were taken. We looked at plant and root biomass, fertile seed number and weight, sterile seed number, which also include the number of sclerotia, as sclerotia replace seed. We also looked at seed germination rates, so the progeny of these infected plants. And then we also took sclerotia weight per individual plants. We found that effects of Claviceps purpurea infections on host physiology ranged from negative to positive depending on disease severity, the plant species, and the plant tissue type observed. In barley, we found significant increases in plant fertility and the average number of seeds per tiller at the 5% infection rate. We also saw significant increases in plant biomass at the 15% infection rate and significant increases in root biomass at the 15% and 25% infection rate compared to controls. These increases can most likely be attributed to the reallocation of host resources to uninfected parts of the plant. For example, since we only infected the main or primary tiller of each plant, the increases we observed in overall plant fertility are most likely attributed to the increased fertility of the uninfected secondary tillers. We also observed the potential for negative effects of Claviceps purpurea through a trend of decreased seed germination rates as infection rates increased. However, there were no significant differences observed. In wheat, we generally saw neutral effects. However, it should be noted that we were only able to utilize a significantly smaller sample size due to the difficulties of achieving the desired infection rates with Claviceps purpurea. However, these results can indicate the potential for variable interactions of Claviceps purpurea infection between different plant species. To begin Objective 3, I'd like to first give some phylogenetic background into the Claviceps genus. The Claviceps genus is divided into 4 sections. First is section Citrina, which is estimated to have diverged around 60 million years ago, and is thought to have one lineage. This lineage has a restrictive host range and can only infect grasses belonging to one grass host tribe. The second section, section Paspalorum, is estimated to have diverged around 57 million years ago and is thought to have 2 lineages. However, one of these lineages is still unknown and is not taxonomically classified. Similar to Citrina, section Paspalorum has a restricted host range and can only infect grasses belonging to one grass host tribe. Next is section Pusilla, which is estimated to have diverged around 50 million years ago and contain around 28 different lineages. These lineages have increased host range potential and can infect grasses from 5 different grass host tribes. And lastly is section Claviceps, which is thought to have diverged around 20 million years ago and contain around 17 different lineages. Although recent research may indicate that the number of lineages in section Claviceps may actually be higher than this. Although section Claviceps is known to have a further increased host range potential, where species within the sections can infect grasses on up to 13 different host tribes. For example, Claviceps purpurea is known to infect up to 400 different grass species across 6 different grass host tribes. As you can see, there are some clear, distinct differences between these sections, such as the number of lineages, or speciation events that have occurred within these different sections. In addition, we also see differences in the host range potential of these sections. Whereas in section Citrina and Paspalorum, we have a very restrictive host range that can infect grasses of only one host tribe. Whereas in Pusilla, we see increased host range where these species can actually infect grasses of up to 5 different host tribes. And then further increased host range of section Claviceps, where these species can infect up to 13 different host tribes. We also see differences in the types of grasses that these sections can infect, whereas in section Citrina, Paspalorum, and Pusilla, these species are only known to infect warm season grasses, while section Claviceps can infect cool season grasses. Therefore, we tried to use Objective 3 to identify some of these genomic differences that we observe between these sections. To identify factors associated with the differences that we observed between the 4 different Claviceps sections, we looked at the genomic architecture of these different species to gain insight into the evolution of the genus and these Claviceps species. For a brief background, plant pathogens can have their genomes classified in 2 different ways. The first is to have a one-speed or a one-compartment genome, which is often considered to be less adaptable. Or genomes can be more aligned to a 2-speed or 2-compartment model, which is often considered to be more adaptable. To classify the genomic architecture within Claviceps species, we looked at several aspects. The first is to look at transposable element, or TE content. For 2-speed genomes are generally considered to have lower or relatively lower amounts of transposable elements within their genomes, while 1-speed genomes are typically considered to have increased levels of transposable elements. We also looked at the TE localization. So where are these TEs located within the genomes, and are they co-localized to certain gene types? For example, in 2-speed genomes, effector proteins or genes that are associated with pathogenicity or virulence might be co-localized to these effector or TE-rich regions. Whereas in a 1-speed genome, these TE-rich regions could have effector proteins located around them, or also just core non-virulence genes. We also looked at genome compartmentalization. This is a synonym for gene density. So where are the genes located, and what are the intergenic flanking region sizes of these different genes? So in a 1-speed genome, these genes have relatively similar intergenic lengths, which means they're found in more gene-dense regions of the genome. However, in a 2-speed or 2-compartment genome, you can see a clear distinction between different genes, where these genes have smaller intergenic regions and are in more gene-dense regions of the genome, whereas these genes are located in more gene-sparse regions of the genome because they have longer intergenic lengths, most likely due to their association with transposable elements. In addition, where are effector proteins or virulence factors found within the genome? Are they in more gene-dense regions of the genome or in more gene-sparse regions of the genome, where these different groups might actually have genes that are evolving at different rates? Next, we looked for the presence of repeat-induced point or RIP mutation. So RIP is a mechanism that is used by fungal species to silence duplicated or repeat sequences. So if this gene duplicated itself, RIP can be triggered during sexual recombination to silence these repeats through C to T mutations, thereby creating 2 mutated sequences that are no longer expressed and are inactive. And lastly, we looked at the presence of gene duplication. For in this diagram here, gene duplication is shown to be part of a one-speed genome. However, in this analysis here, gene duplication is seen to be a method for one-speed genomes to be adaptable. So if this is a method for one-speed genomes to be adaptable, the process of gene duplication can also be considered to be part of the two-speed genome model, which is thought to be part of more adaptable organisms. To classify the genomic architecture, we first looked at transposable element content. We did this by plotting the lengths of transposable elements within each of these species to get a sense of how much of the genome is composed of transposable elements. So for section Claviceps, these species averaged around 8.8% of their genome composed of transposable elements, which is relatively low compared to section Pusilla, which had around 32.5%, and section Paspalorum, which had 17.5%, and further still section Cichina, where it had 51.7% of its genome composed of transposable elements. Next, we looked at transposable element localization. We did this by identifying the average distance of genes to transposable elements. And what we found is that in section Claviceps and in section Pusilla, effector genes were significantly located closer to transposable elements compared to all other genes within the genome. And this is not the case for sections Cichina and Paspalorum, where transposable elements were more dispersed and not co-localized around genes of certain types. Next, we looked at genome compartmentalization, and we did this by plotting the 3' and 5' intergenic flanking regions of each gene. What we see here is that all species of Claviceps fit to a one compartment model. However, if we looked at where effector proteins are located, we can see that effector proteins actually have significantly longer intergenic flanking regions in section Claviceps and in section Pusilla. We found no significant differences between effector or the intergenic regions flanking effector proteins compared to other genes in section Paspalorum and in section Cichina, which then indicates that effectors are located in gene-sparse regions in section Claviceps and Pusilla, while they're located in more gene-dense regions in Paspalorum and Cichina. Next, we looked at the presence of RIP, and this is done through scanning the genome in windows of around 1,000 base pairs to get a RIP composite index. So RIP composite index indicates that if you have a positive value, these windows are thought to be affected by RIP, indicating that RIP is active in those regions, whereas a negative RIP index indicates that RIP is inactive and that RIP is not even present. These also show that from the program we use, we can identify the percent of the genome that is affected by RIP. And clearly we see here that in section Claviceps, RIP is inactive and is not working, whereas in section Pusilla, Paspalorum, and Cichina, RIP appears to be present. And lastly, looking at gene duplication. So this was done by blasting the proteins in each species genome against itself to identify protein pairs or gene pairs that have greater than 80% identity. And those are classified as a gene duplicate. What we see is that in section Claviceps, we have upwards of 400 to 500 duplicated genes within each of these species genomes, whereas this is significantly reduced in section Pusilla, Paspalorum, and Cichina. And this is a further indication that RIP is absent in section Claviceps, as RIP would be working to try to silence these gene duplications as it appears to be, or as it appears to be doing in section Pusilla, Paspalorum, and Cichina. In conclusion, these results of genomic architecture within the Claviceps genus has indicated that the Claviceps genus most likely started with specialists that have small host range, that have aspects of a more one-speed or less adaptable genome model of section citrina and section passiflorum. Through evolutionary time, these genomes evolved towards a two-speed or more adaptable genome, where we also saw increased levels of speciation and increased levels of host range potential of these Claviceps species. And this is most likely due to the co-localization of transposable elements around effector proteins, so virulence factors or predictive virulence factors, as well as effectors being in gene-sparse regions of the genome, where they could have different mutation rates than the core genes that are found in gene-dense regions of the genome. And lastly, we see evolution towards an even more two-speed genome model, where in section Claviceps we further saw increased host range potential, and section Claviceps also had a lower TE content. RIP was found to be inactive, which most likely resulted in gene duplication, potentially providing increased adaptability to these species in section Claviceps. To gain further insight into the evolution of Claviceps, we wanted to take a closer look at Claviceps purpurea. To do this, we identified and constructed the pangenome of Claviceps purpurea and created the positive selection landscape. But first, I wanted to give a quick background onto what a pangenome is and to what selection is. So a pangenome is the entire pangenome of a species that is constructed from multiple isolates or strain of that species. It's composed of a core genome of genes that are shared by all members or all isolates within that species, and an accessory genome of genes that are not shared by all members. Pangenomes can be classified as a closed genome, where they have a larger core genome and smaller accessory genome. And this is more often found in organisms that are a bit more specialized in nature. Or pangenomes can be more open and is often found in more adaptable organisms, as they have this larger accessory genome and a smaller core genome, which allows these organisms to have a more global distribution and to inhabit different environmental or ecological niches. In accessory genomes, pangenomes have been a bit more studied in bacteria, where the accessory genome can range from either 2% or 95% of the entire pangenome. Fungi are a bit more understudied, where 4 pangenomes of fungal organisms have identified that the accessory genomes can range between 9 to 19% of the entire pangenome. However, recently, Zymoseptoria tritici, similar to Claviceps, is a biotrophic fungal pathogen of grasses, most notably wheat. And this species was shown to have an accessory genome of 40%, so relatively large compared to the other fungal species that have been classified. Next is selection. So for selection, you can either have negative selection, and this is an example of stabilizing selection, where the organism is actually trying to purify or fix amino acid changes. Where here, if you have the original population and then population after selection, this organism is trying to maintain its genes the way they are because they're effective and they work and they don't want to be changed. Or you can have positive selection, where the organism is actually trying to favor amino acid changes in one direction or the other, so that that gene changes its functionality and provides a fitness advantage to the organism. Results from the Claviceps purpurea pangenome revealed that this pangenome consists of 10,540 orthogroups, or genes. 62% of these genes were found to be core genes that are shared between all of the 24 isolates that we utilized, whereas the remaining 38% of genes were found to be accessory and were found to be in anywhere from 23 to individual isolates. This core genome was found to be enriched in carbohydrate active enzymes, or CASEs, as well as peptidase proteins, transmembrane proteins, and proteins with conserved domain functions. And this is very concordant with results of other fungal pangenomes. The accessory genome was found to be enriched in genes that are more associated with pathogenicity or virulence, such as genes that have secretory signals or genes that are predicted to be effectors. However, we also found that the accessory genome was also enriched in many genes that had unknown or unclassified function. And lastly, we found that secondary metabolites, which are a big component of Claviceps purpurea, were not found to be significantly enriched in any of the three pangenome categories, indicating that these genes are a bit more dispersed and variable between individual isolates. Moving on to the positive selection landscape of Claviceps purpurea. This analysis was done through the program PAML with the associated algorithm CodeML to look for signatures of positive selection at individual codons within the gene. The input into this program was 6,244 single copy core genes, indicating that this analysis can only be conducted on genes that are shared between all isolates and thus do not represent genes from the accessory genome. From this analysis, we identified 986 genes with signatures of positive selection at an alpha of 0.01. Most of these genes were genes that had conserved protein functions or were unclassified or had unknown functions. It was very interesting to observe only 2 predicted effector genes had signatures of positive selection. Looking at the proportion from input, we see that secondary metabolite genes and unclassified genes had the highest number of genes with positive selection based on the number of those genes in the input dataset. And again, we see that lack of effector or low number of effector proteins with signatures of positive selection. This is kind of contrary to some of the hypotheses around plant pathogens, and mostly the Red Queen hypothesis, which dictates that organisms must continuously adapt and evolve in order for species to maintain its relative fitness among the system that is co-evolving with. Plant pathogens co-evolve with its hosts because plant pathogens are continuously trying to evolve and adapt its virulence or effector genes so that it can actually infect the plant. Whereas the plant is continuously evolving its genes to provide defense mechanisms to not allow the pathogen to infect it. Since the plant is continuously evolving these defense mechanisms, you would expect to see continuous adaptation and evolution of these predicted effectors or virulence proteins so that the pathogen can continue to adapt this plant and defeat those newly created defense mechanisms. However, this does not appear to be the case for Claviceps purpurea, indicating that these effectors are not under selection or not under positive selection for adaptation. If we look at the number of codons per gene that are under positive selection, while not significant, we see that secondary metabolite proteins had the highest number of codons per gene with signatures of positive selection, which could indicate that secondary metabolite proteins are more important for the evolution and adaptation of Claviceps purpurea compared to effector proteins. In conclusion, we found that Claviceps purpurea has a relatively large accessory genome that is enriched in genes related with pathogenesis, such as genes with secreted signals or are predicted to be effectors. We also identified that many of the core genes of the Claviceps purpurea pangenome have positive selection on secondary metabolite genes. Secondary metabolite genes can often convey infection potential or virulence of the pathogen. However, secondary metabolites often convey stress tolerance and provide a fitness advantage to the fungal organism, such as against UV radiation, as in the production of melanin, or stress tolerance for oxidative stresses or colder climates. If we recall earlier from the Claviceps genus, section Pusilla, section citrina, and section paspalorum are all known pathogens of warm season grasses and more often inhabit warmer climates that are often more shady. However, section Claviceps, these species infect cool season grasses and are often found in more colder climates that are more open to the elements, potentially indicating why we see this positive selection on secondary metabolites in Claviceps purpurea, which is a part of section Claviceps. In addition, recent research has identified many cryptic species of Claviceps purpurea sensulato, which is the pre-molecular concept of Claviceps purpurea. These cryptic species reside in section Claviceps, and the divergence of these species have been associated or related to adaptation to different ecological niches, such as some species inhabiting more wet and open environments, while others are found in more coastal environments, and others are found in more mountainous environments, whereas Claviceps purpurea is a bit more broad and can be found on grass species in many of these different environments. Together, this large accessory genome, enriched in pathogenesis-related genes, as well as this positive selection on secondary metabolites, which might be providing increased fitness to Claviceps purpurea through stress tolerance, such as colder climates or adaptation to ecological niches, could indicate or provide factors associated with the broad host range and global distribution of Claviceps purpurea. Lastly, I'd like to go over the future direction of Claviceps research, which should focus on studies examining the potential for a co-evolved symbiosis with rhizotomous grasses. This theory can be extrapolated from the three objectives discussed in this presentation. In the first objective with the field surveys, we identified that brome species were highly infected or are highly susceptible to ergot infections. Brome species generally have low seed set, which means that around 30% of its florets get pollinated and turn into seed, leaving around 70% of these florets open for Claviceps infections. Brome species are also highly abundant as they are invasive species and are highly rhizotomous, meaning that they proliferate greatly through asexual reproduction through rhizomes underground. In Objective 2, with the greenhouse inoculations, we identified that higher infection rates of Claviceps purpurea on hosts showed increases in root biomass, which could be correlated to increases in rhizotomous biomass as well, indicating a potential for increased asexual reproduction due to infection. In addition, higher infections also showed decreases in seed germination rates, effectively lowering the sexual reproductive potential of hosts due to Claviceps infections. Taken together, this potential increase in asexual reproduction and decrease in sexual reproduction indicate lower genetic diversity of hosts due to Claviceps infections. In Objective 3, with the genomic analysis, we identified that effector proteins were not under positive selection, which could be associated with this lower genetic diversity of hosts. This lower genetic diversity could potentially be slowing down the evolution of host genes to combat the infection of Claviceps. This evolutionary slowdown of host defenses mechanisms could help explain the observations of a lack of positive selection on effector proteins. In addition, the observations of grazing avoidance of infected grasses indicate the potential for a conditional mutualistic relationship between Claviceps and its hosts, as infected plants have higher fitness due to not being grazed. Through a simple illustration, we can gain insight into what this co-evolved symbiosis might look like. This can be examined through a scenario where we have populations of highly susceptible rhizonomous grasses and populations of low susceptible grasses at sites where there is no ergot presence, the presence of ergot, and the presence of ergot with mammalian grazers. During infection, these highly susceptible rhizonomous grasses will most often be highly infected. Following this population into the next year could show indications of a change in host population structure through the reduction of low susceptible grasses and increased abundance of highly susceptible rhizonomous grasses. Continuing the cycle will further elucidate this trend. While initially highly rhizonomous grasses might have initially higher fitness and invasiveness potential, these ergot infections might actually be increasing the fitness of these hosts, particularly if grazers are present. While this shows the potential for increased fitness of these highly susceptible rhizonomous grasses, this also ensures the abundance of highly susceptible grasses to guarantee the continual propagation of ergot. In the end, I would like to thank all of those involved, particularly Judith Jolly for her assistance in conducting the ergot field surveys, as well as the San Luis Valley barley growers for allowing me access to their fields. I would also like to thank Miao Lu for her assistance in obtaining raw reads of up to 30 Claviceps genomes, as well as Miroslav Kolarek for access to additional Claviceps species for sequencing, and allowing me to travel to Prague for 2 weeks to learn how to analyze ergot alkaloids through high-performance liquid chromatography. Additionally, I'd like to thank all members of my committee, as well as my research associates in the Broders Lab, Nalem Lab, and the Chapa Lab. I'd also like to thank Stephen Mondo for his assistance in understanding different genomic analyses, as well as my friend Rick Emery for his assistance in answering my many Python-related questions. Lastly, I would like to thank all of my funding sources for providing the means to conduct this research. Thank you for listening.